distinguished panelists and all attendees to today's webinar. Good evening, good morning, and good afternoon. This is Meng from the Neurodegeneration Research. It's really great honor to have two amazing speakers today for novel pathological mechanisms for neurodegeneration. So I just would like to take the next 3 to 5 minutes for a brief introduction on the backgrounds about our series webinar. So the whole NRR series webinar is hosted by Neurodegeneration Research, and the journal editor-in-chief is Professor Kuang-Fu Su from Jinan University, and the journal is aimed to explore new horizons in neurodegeneration. And we publish the outstanding articles and reviews in neuroinjury, neural repair, and neurodegeneration. So we are very glad to announce the latest impact factor of the journal has reached to 6.1, ranking top 15% among 272 journals in neuroscience. And we would like to acknowledge our contributions from our editorial board members, our reviewers, our readers, and most and certainly not the least, our authors who have chosen to submit their excellent articles to neurodegeneration research. So just in 2023, in the past year, we published over 514 articles, including 38% of them are our original articles and 27% of them are review articles. And we also publish nearly 1/3 of all our papers as editorial materials. So you'll be interested to look at our perspective papers that who are mostly written or published from the authors outside China. And along over 2,000 authors published papers with us in 2023. We published from 48 countries. And we publish papers outside China is over 70% of all these papers we publish. And also in 2023, in EICI, which is the Essential Science Index, just in 2023, in the last past year, we have over 180 highly cited papers, which means top 1% highly cited papers in neuroscience. And we also publish 15 papers as hot papers, which is top 0.1% papers in neuroscience, so which is very impressive. And just regarding the annual conference we hold, as the brand name of Neurodegeneration Research is our International Neurodegeneration Symposium, which was started since 2011, and this is our 12th year. And last year was held in Chengdu, and this year the symposium will be held during April 12 to 13 in Guangzhou. So welcome to meet face-to-face during the symposium. And just starting from the March 2021, which during the pandemic, our journal has conducted several series online meetings, which is our free series webinars with all these distinguished experts and also our best performance authors from all over the world. Just in 2023, we hold 11 webinars with total 165,000 views just in 1 year. So today, it's very honored to have these two amazing speakers with us who will present the topic in novel pathological mechanism for neurodegeneration. And first, I would like to introduce today's moderator and the session chair, Shangshan Dai. Professor Dai is from Department of Biochemistry and Molecular Biology of Army Medical University from China. She is an active researcher and an expert reviewer for many important scientific research projects in China. And Professor Dai's area of interest focus on the immunological and metabolic regulatory mechanism of brain injury and repair. And she published many impressive research results, including brain behavior immunology, sense advance, and etc. And her work has been published in F1000 research and been cited by top scientific journals. So next, I would like to hand over the host to Professor Dai for the following scientific sessions. And thank you for paying attention to our series webinar.
Okay, thank you. Thank you, Mom. Okay, it's my turn, right? So
Yes.
Okay, thank you. So welcome the professors, researchers, and scientists. It's my honor to be the chair of this webinar. So first, let me briefly introduce our two amazing speakers tonight. The first one is David Campanelli. He is the principal investigator of Department of Diseases and Hormones of the Nervous System of the Paris Saclay University, France. So the Dr. Davis focused on the research programs focused on aging and neurodegenerative diseases. Using multimodal approaches from cell biology to behavior, they studied key mechanisms to discover novel pathways, in particular on protein clearance and the role of synaptic activity. Their ultimate goal is to validate novel therapeutic strategies for patients. So welcome, David. The next amazing speaker is Marina Garson-Marshall, and she is from the Raymond Cajal Researcher Institute of Functional Biology and Genomics, University of Salamanca, CSIC, Spain. So her speech topic is impact of fatty acid on astrotic bioenergetics. Her lab is interested in lipophage role in physiology and diseases. Particularly, they are focusing on macrophage differentiation, such as M1 to M2 transition, and how lipophage can modulate these changes. The metabolic phenotype and cytokine profile in both types of activation, such as M1, M2, are quite well known. However, the molecular process that triggers the switch remains obscure. They are trying to provide insights to unreveal the required molecular and cellular changes to switch from M1 to M2 macrophages, which will make an important impact in the field. Thus, it will point to novel targets to modulate immune activation, which will provide the basis for treatments for such as NASH, NAFLD, liver fibrosis, and obesity. So it's my also honor to introduce the amazing planets of tonight. So I think tonight is very beautiful and exciting. The first one planet is Peng Shu Wei, the director of Department of Rehabilitation Medicine, National Research Center for Rehabilitation Technique, AIDS, China. Dr. Wei's research interests focus on strategies for screening and prevention of AD and methods for cognitive training and rehabilitation, including applying non-invasive neuromodulation treatments in patients with neurological diseases. And the second scientist and professor is Chao Yan. He is the chairman of Department of Physiology, Nanjing University of China. He got his BS in biochemistry from Nanjing University in 2003 and PhD in toxicology from the School of Pharmacy at the University of Colorado in 2011. He then received postdoctorate training at the University of Colorado Cancer Center until he came back to Nanjing University in 2016. His lab studies the pathology and pharmacology of neurological diseases, including epilepsy and glioma. He has a particular interest in glial biology and lipid metabolism, and has published many high-impact articles in prestigious journals such as Nature, Nature Neuroscience, Cell Research, GCI, and Cancer Research. So he's an amazing and very exciting scientist and professor. Welcome, Yan. Okay. According to the schedule of tonight's webinar, then let's welcome the first speaker, David, bring his topic, that is protective mechanisms of neuronal stimulation in AD and tauopathies. Welcome, David.
Okay, hello everybody. Well, first of all, thank you very much for inviting me to give this talk. I assume everybody listens to me and everybody can see my screen, hopefully. We tested before. Okay. So,
yes.
All right, perfect. So I'm very glad to be here. Thank you for inviting me. And yeah, so the schedule is pretty tight, so I'm gonna start right away with my presentation. As mentioned, I've been working on Alzheimer's disease for over 20 years, in particular the pathology of synapse. And during the last let's say 10 years, I focused more on the protective effects of synaptic stimulation against pathological tau. So I'd like to start with this image, which shows a painter was called William Utermohlen, who was diagnosed with Alzheimer's disease back in 1995. And the particularity of this author that he did many portraits during his life, and through the portraits that he started to paint when he was diagnosed with Alzheimer's disease, we can really immediately see the dramatic progression of the pathology in his portraits. And the author did in 2007. So Alzheimer's disease is a very drastic pathology which affects ultimately the whole brain. Aging is the main risk factor. So for countries where the expectation of life are increasing, like Europe, China, or United States, Japan, of course, is a pathology that is becoming more and more common, with about 46 million people worldwide that are already sick, and a projection of more than about 130 million around 2050. So it's pretty dramatic. So it's something important that we find a therapy for this increasing disorder. Now, from this pathological point of view, many of you probably know, but the pathology consists of neurodegeneration, progressive neurodegeneration, which affects ultimately the whole brain cortex, starting from the temporal lobes, and in particular the hippocampus, where memory formation occurs, and then it spreads through to all the frontal cortex, parietal cortex, and ultimately also to the occipital cortex. And the two main histopathological hallmarks are the position of beta amyloid intraneuronally and also in plaques, and aggregated tau. Now, today, as mentioned, I'm going to focus more on the tau pathology. So tau is one of the microtubule-associated proteins stabilizing microtubules in neurons, in particularly mainly in axons. And tau detaches from the microtubules when it's phosphorylated. So during Alzheimer's disease and tauopathies, this protein becomes hyperphosphorylated pathologically, and it aggregates, generating toxic oligomers and then ultimately tangles. Now, a recent study showed that the tau pathology is associated with reduction of synaptic function and also synapse loss in Alzheimer's disease patients. In this study, they show by magnetoencephalography, this is in a live patients, a small cohort, about 10 patients, they show that there is an inverse correlation between the accumulation of tau that was detected with a particular marker for tau, and magnetoencephalography, which basically measures the neuronal activity, and as well with the loss of synapses. So this is a marker that was used to visualize in alive patients' synapses, and this is again tau. And we see that there is again an inverse correlation. So the more tau we have, the less synapses we have. And this is very interesting because it is known that synapses are a crucial target in Alzheimer's disease. In fact, it has been known for years that synaptic marker proteins like this VGLU1 are reduced in Alzheimer's disease, as well as total synapses. And most importantly, we know that loss of synapses is the best correlate with cognitive decline in Alzheimer's disease patients. Now, again, through new technology and PET scan, we can now see this loss of synapses occurring in Alzheimer's disease patients alive, as you see here in red, that is decreased in Alzheimer's disease people. And of course, loss of synapses have been detected in many different models, in vivo models and in vitro models of Alzheimer's disease. Notably, in mice, typically the neurodegeneration is not that dramatic, but we see the memory loss, which is mainly associated with the loss of synapses. So our group and other groups demonstrated that synaptic stimulation, I'm talking about a physiologic synaptic stimulation, of course, not like a massive depolarization or like a teratogenic stimulation of neurons. So physiologic synaptic activation can be protective against A-beta toxicity. Epidemiological studies also show that higher educational attainment or the participation in intellectually stimulating activities also is associated with reduced risk to develop Alzheimer's disease. Besides synaptic activities important for neuronal survivors, it is involved in activation of survival pathways. Ultimately, it has been found that deep brain stimulation of Alzheimer's disease patients can result in positive outcomes, such as recovery of hippocampal volume for certain patients, increased brain metabolism, and also better outcomes in cognition and memory. So just a little introduction. Deep brain stimulation is a surgical technique used to treat neurological but also psychiatric disorders, such as depression or even morbid obesity. And it consists of the implantation of an electrode in a very specific area of the brain. Then there is an extension cord which gets to a internal pulse generation, usually subcutaneously. And here we can basically induce electrical activation of the electrode with deciding the frequency and the time lapse of the electrical stimulation depending on the different pathology. For Alzheimer's disease, the area that is interesting that came out quite recently is the fornix, which is part of a larger circuit involving memory formation called the circuit of Papez that goes from the entorhinal cortex to the hippocampus via the perforan path, then the exit from the hippocampus via the fornix to reach the mammillary bodies, and then again in other areas to go back to the entorhinal cortex. So deep brain stimulation of the fornix influences brain structures in Alzheimer's disease. Like here we have a control patient, after control patients actually, it's an average of 25 patients. They have like decrease in yellow brown, means areas of decreased volume. While after 1 year of deep brain stimulation, certain patients could find even increase in hippocampal and parahippocampal areas, which was a remarkable result. So from our perspective, we wanted to see what are the cellular effects of deep brain stimulation and synaptic activity in general on neurons and proteins involved in Alzheimer's disease. To do so, we use an in vivo model, which is the triple transgenic mouse model for Alzheimer's disease, which carries 3 mutations, one for APP, one for presenilin-1, and the other one for tau, and develops both pathology, meaning the A-beta pathology and the tau pathology. In this mouse, we did an implant of 2 electrodes, so bilateral implant of electrodes in the entorhinal cortex, and we did stimulation with the protocol that is shown here for 25 days. Then the mice went through a battery of behavioral tests, and then they were euthanized at 7 months for biochemical and histological studies. So from the histological point of view, we could see here, we have a wild-type non-stimulated mice, so that are the mice sham who received the surgery with the electrodes but were not stimulated. Wild-type stimulated mice, transgenic non-stimulated mice, and transgenic stimulated mice. So the markers that we did were for synaptophysis, so a synaptic marker, presynaptic marker, and for the tau oligomers, so for aggregated soluble small molecular weight tau aggregates. So we already see that the transgenic has less synaptophysin compared to the wild type. But if we check at the stimulated group, we see that there is a recovery of synaptophysin back to the wild type levels. Concerning the tau pathology, so the tau oligomers were not present as expected in the wild type mice, but they were present in the transgenic mice. But again, with the stimulation, we had a reduction of toxic oligomeric tau in terms of puncta, area, and also as intensity of the staining. We also tested for ATA, which is like the most common hyperphosphorylated form of tau, pathological form of tau, also used for diagnosis. And we can see the same results. So a reduction of this hyperphosphorylated tau in stimulated mice. Then we did the contrary. So we wonder what happens if we do a chronic inhibition of activity. So in order to do so, we use another mouse model, which is for PS19, which overexpress a human tau harboring a mutation for a familiar form of frontotemporal dementia. And this mouse model was developed by Virginia Lee in Pennsylvania. Already show a progressive accumulation of tau already at 6 months, and a loss of synaptic proteins, like here is a synaptophysin, progressively gets reduced in the PS19, and also postsynaptic markers. Here is a GluR23. And of course, also a loss of synaptic plasticity, so with memory deficits. In order to do a chronic inhibition, we perform a whisker differentiation unilateral, so just on one side of the snout. And this, so the whisker, you know, is very well connected to the somatosensory cortex called the barrel cortex, because neurons here are organized into barrels, like the clusters that are specifically connected to each whisker. And so the whisker through the brainstem thalamus, there is the somatosensory cortex. If we take out the input, which are the whiskers, it will result in a decreased activity in the barrel cortex. So we did a surgery, so we're removing even the whisker bones, so no whisker growth. So this is 5 months inhibition here, so no whisker to go back. And we did a cytochrome oxidase staining just to see that indeed there was like a reduction of activity in the deafferented cortex. Now, to be noted, this is the same brain, so we have the control side and the deafferented side. Here you can see the barrels of the somatosensory cortexes here are less stained. And the beauty of this technique is that it's not invasive, because we just make a surgery on the outside. We don't use drugs that will alter metabolism, and we have the control in the same brain. We repeated the staining that I showed before on the triple transgenic against synaptophysin. You see already in the transgenic mouse, we have a loss of synapses, so a synaptophysin marker compared to the wild type control. And in the deafferented side, so this is the same brain, so control side, deafferented side. In the deafferented side, we could see that there is a further reduction of synaptophysin, which was not because of the deafferentation, because we see that in the control, so in the wild type mice, despite the differentiation, this side of the barrel cortex still maintains the staining of synaptophysin. Concerning the oligomers, we saw the opposite with the stimulation. So we saw an even more important deposition of accumulation of tau oligomers. And the same occurred for the 88 immunostain. So this is interesting because it was in even in vitro models, but we wanted to know what happens in the humans with the brain stimulation. And since the cohort now, the brain stimulation of the fornix is a collaboration I'm having with Professor Andres Lozano in Toronto. It's in the phase 2, and we don't have brains from patients that are still alive. I wonder where we could get regions that were stimulated with deep brain stimulation. And of course, deep brain stimulation was used for Parkinson's disease treatment. And we know now that tau accumulation is observed more and more in Parkinson's disease patients. So we could get samples of Parkinsonians who were stimulated with deep brain stimulation. And we got like a cohort of controls and a group of stimulated Parkinsonians. So we check in the cortex and we could see that the two groups had comparable amount of tau pathology in their brains. Then we know that to treat Parkinsonians, typically the stimulation occurs in the subthalamic nucleus, which project more than 90% of its axons into the inner globus pallidus, so which is the region right here. It is exactly in this region that we went to see what happens to tau pathology. So we are in the inner globus pallidus. You can see the control that we have somas that are positive to 88, so positive neurons to tau pathology, and a good amount also of neurites containing tau. In the deep brain stimulation group, we couldn't find any positive somas, which was a pretty remarkable result. And we actually could see an increased density of neurons, so showing that the stimulation was neuroprotective. We then investigated the molecular mechanism behind tau clearance. To do so, we use an in vitro model consisting of primary neurons from, I'm sorry, here there is a mistake, just disregard this 2576 from the PS19 mice. And we could see that in vitro, these accumulate progressively pathological tau, and they have a reduction of synaptic proteins. Again, this is postsynaptic density 95. These neurons in culture, so 2 weeks already, they also show accumulation of tau oligomers. And if we stimulate these neurons with a protocol that involves the activation of NMDA receptors, mimicking long-term potentiation, we see a reduction of the tau oligomers as well as the hyperphosphorylated tau. In this model, we see that this degradation, this reduction of pathological tau requires lysosomes. In particular, with stimulation of synapses, we have an increased activity of lysosomes. This is an in vivo experiment, live imaging experiment, where we feed the cells DQ-VSA. It's a particular VSA that fluoresces when it gets cleaved, and typically is transported to lysosome for degradation. So the more fluorescence we see, the more the lysosome are active. And with the stimulation, we see increased lysosomal activity. Since lysosomes are involved, we decided to investigate the transcription factor AB, which is a pivotal player in lysosomal biogenesis and autophagy. TFAB is a transcription factor that is phosphorylated to keep it inactivated and in the cytoplasm. When activation occurs of TFAB, it gets dephosphorylated and it gets it traffics to the nucleus to start the transcription of its downstream genes, which involve, for example, subunit of the lysosomal proton pump or BATP, or also, for example, P62, that is a protein involved in autophagy. And in the literature, we found that increased expression of TFAB protects against models of frontotemporal dementia, Alzheimer's, and also motor neurodegeneration. TFAB mediates tau clearance also. This is a more recent paper via the lysosomal exocytosis. So it could be another mechanism triggered, for example, by synaptic activity, and prevents intraneuronal tau pathology and spreading. So we're intrigued by the involvement of TFAB. And now I make a long story short, but what we did was to, first of all, transfect neurons with a TFAB GFAP, and then see what happens to GFAP during with a live imaging time course. And we could see that progressively, like in about 20 minutes, we could see that the fluorescence, so TFAB was going from the from the cytoplasm within the nucleus. So it was getting activated. Of course, there was an overexpression of TFEB. So we also demonstrated that biochemically by purifying nuclei or separating nuclei and cytoplasm. And we could see that in steady condition in controls, we have more TFEB in the cytoplasm rather than in the nucleus. But with the stimulation, we have the other way around. So we have reduced TFEB in the cytoplasm and more in the nucleus. And this is endogenous TFEB. And these are the histonic proteins showing that it is a nucleus-enriched fraction. As I mentioned, TFEB needs to be dephosphorylated. So we also check one phosphorylation site on the TFEB, which is the serine 142. And with stimulation, we could see that there is a reduced phosphorylation of TFEB. So meaning stimulation activates TFEB. We checked by real-time PCR the downstream genes of TFEB, and today I'm going to focus in particular on this subunit, the ATP6V0D1, is a subunit of the VTPase proton pump, which contributes to the acidification of the lysosome and therefore to its correct function. And so we could see an increase in the RNA with stimulation, and we check this is in primary cultures, primary neurons. We could see an increase of this subunit with stimulation in neurons. And then we went to check this in the triple transgenic mouse model that I showed you before. And already in the transgenic non-stimulated, we have a decrease of this subunit compared to the wild type. With the stimulation, we have the recovery of this subunit. And not only the subunit is increased, but it also localized more into lysosome. So you see here that the protein is depleted in the transgenic. With the stimulation, we have the recovery of it, but we also have a recovery in the colocalization of this subunit within lysosome. So supporting the idea that the lysosome are more functional and more active in the degradation of pathological tau. So to summarize what I told you so far, we saw the synaptic activity reduces pathological tau species and restores level of synaptic proteins in wild type to wild type levels in models of tauopathies. Very interestingly, DBS reduces phosphorylated tau and prevents neuronal loss in Parkinsonian patients. The tau clearance during synaptic stimulation requires the lysosomes and their degrading activity. And finally, the synaptic stimulation activates TFEB and the expression of its downstream genes, which are required for tau clearance and enhancement of lysosomal function. So to conclude, I want to acknowledge the several collaborators that helped me in putting together this data. Of course, Yvette Aqua and Professor Beaulieu from our institute here in, I think, CERN. Professor Andres Lozano that I mentioned before at the Division of Neurosurgery at the Western Hospital in Toronto, Canada. The precious collaboration of Chiara Di Malta and Carmine Settembre, the Teleton Foundation in Pozzuoli, which helped me with the TFEB project, the expert of TFEB. My ex-colleague and valuable pathologist Estibalis Capetillo Zarate at the University of the Basque Country in Bilbao, helped me with the staining of the Parkinsonians. And then Rakesh Kayat and Jeffrey Cordover for providing me the first with the T22 antibodies for tau oligomers when it was not commercially available yet, and Jeff Cordover for the Parkinsonian brains. And thank you very much for your attention.
Okay, thank you, David. Thanks for your perfect presentation and thanks for such a beautiful work. So after the second speaker finished his presentation, we're going to the Q&A session, right? Okay. Let's welcome the second speaker, Marina Gasher-Michel. Her topic is impact of fatty acids on astrocytic bioenergetics.
Right.
Welcome, Marina.
Hi, thank you so much, and thank you so much for the kind introduction. Are you hearing me well? So,
Yes,
okay. Thank
can
you,
hear
thank you.
very
And the screen is already sharing, I guess. If not, just let me know. So, well, today I'm going to explain a little bit about my last project as a senior postdoc in my previous lab. Now I just opened my own lab, so that's why the explanation that you provide was about macrophages, but I'm still doing a neuro research line, so I didn't abandon that. So, well, I guess this is not, I shouldn't be explaining that in our brain we have many different types of cells, but just a reminder. So, and when we have a lesion or we have to regenerate parts of our nerves, the neurons are important, but the more important cells in this kind of neuroregeneration are oligodendrocytes and also astrocytes. And what I read recently is that if you try to modulate the metabolism, you can improve this regeneration. So that's why I guess my topic today will be fitting very nicely with the journal and with the people that is listening, because what we are in my own lab very, very good doing is understanding the metabolism in neurons, but also in glia, and specifically in astrocytes. And my talk will be about that today. So what is more or less established and we know a lot about is how is the glucose metabolism in the brain. And we know mostly about astrocytes and neurons. And there was explained some theory some years before that now I guess is settled for all the field that what is happening in the brain between astrocytes and neurons in about glucose metabolism is the pyruvate lactate shuttle, so that the astrocytes provide lactate to the neurons, and this lactate will be consumed by the neurons mitochondria to provide the ATP. So in the astrocytes, we know that the glucose will be processed into pyruvate through glycolysis. This pyruvate will be transferred into lactate, and the lactate will be provided to the neurons that they don't do glycolysis that much, and that lactate will be used as a fuel. So that was explained basically by Pellerin and Magistretti a few years ago, and then it was evolving and getting more complete the story. And what it was explaining in my own lab was that the main character in modulate this switch in the glycolysis is the enzyme PFK-FB3 that is mainly expressed in astrocytes, and in neurons is always being degraded, and the glucose works in the neurons to replenish the antioxidant potentials, to replenish the glutathione. So that was explained in these papers that we have here. And now we are just getting into this was mainly proof in vitro, and now we are using the in vivo models to test this in vivo too. And for example, we have a paper in bioRxiv that is under review in a nice journal about how is PFK important in the neurons. So you can sneak a peek and check for the PI in bioRxiv, and you will see what is going on there in vivo. And after learning about all of that, we wonder what was happening about the fatty acids, because we know that the fatty acids and the metabolism is important in the brain too, although glucose is the major fuel. We know that fatty acids are important. We know that the ketogenic diets can be used for therapies, for example, epilepsy. And we have some bit and some clue that the fatty acid metabolism was more important in astrocytes. For example, I like to show always this paper that I love. And in this paper, Maria showed how the toxic lipids are exocytized by the neurons. So the neurons get rid of the toxic lipids, and the astrocytes are able to get them and metabolize them, so we don't have lipotoxicity in the brain. So that and other reports put us in the clue that the metabolism of the lipids in the astrocytes were very important. So we decided to go for it and study that. And we did that through a special mice model. It's we ask for a mice that has a flux CPTA gene. So that is the whole body. So then we modulate these flux mice to get specifically knocking down the CPTA in the astrocytes. But just to explain a little bit, because it's important, why it's important, this CPT1, just like a brief thing. So this CPT1 is very important because it's the channel, the door of the fatty acids into the mitochondria. So we need this CPTA because we need to put inside the mitochondria the long and medium fatty acids. They cannot go through the membrane. And these fatty acids always need to be activated to be able to be transported. And then for this transportation, they need the CPT1. This will put the fatty acids in the intermediate space in the mitochondria. And then we have the CPT2 to put the fatty acids inside the mitochondria. And inside, the fatty acid oxidation can be happening, but we need to put the fatty acids inside. This CPT1 is much more important than the 2 because its function is inhibited by the product or the intermediate that it will produce more long fatty acids and storage the lipids in the cell. So it's a very crucial part in this metabolism. That's why we decide to knocking down this protein to prevent all the lipid metabolism through in this step. And we borrow that mice, we use it to suppress the CPTA specifically, that is more abundant in the brain, and just in the astrocytes. And to do so, we use an in vitro model and an in vivo model with the same mice. For the in vitro model, we prepare primary cultures of astrocytes, and then we suppress the gene using a CRI virus, a general one, and we got the wild type astrocytes and the CPTA1. I need to drink a little bit because I'm getting very dry. And when we did that, we got a very nice result. You can see here the expression of CPT1A, and here you can see the cow astrocytes, and the reduction was very good. For the in vivo system, what we did was inject a virus in the mice, and one with the CRI and the other without the CRI. So we have wild-type mice, and sorry, I need to stop a little bit because I'm losing my voice.
Okay, that's
Sorry.
okay. Relax.
I have a cold, so I'm doing funny things with the voice, so sorry. So again, with the in vivo system, we inject the virus with the CRI. This virus was able to go through the blood barrier, so it was doing what it has to do. And we have the empty virus for the wild types. We generate these mice, we leave them grow, and we have some experiments that they show in vivo, just with the pieces of the brain or the homogenates. And then we have a way to do like a vivo system, isolating by immunomagnetic assay the astrocytes specifically. So that was the ex vivo part of the project. And after having those models, we test again if they were working nicely, and we assay with the whole brain the beta oxidation using palmitic label in whole carbons, and we saw the production of CO2. And with this assay, we could evaluate the beta oxidation and the TCA. And we saw a nice reduction of the beta oxidation in the brains of the CPTA, so the mice was working. And also when we collected the astrocytes, we evaluate the CPTA expression. And in these astrocytes, we saw the reduction of the protein. And in the other cells, the other rest of the cells, there was okay. There was no a reduction. So it was very specific. Then we wanted to go a little bit further, and we did a metabolomics assay, and we evaluate all the fatty acids that they were in these brains, and we saw how the middle and long fatty acids, they were accumulating in the KO mice, while the short ones, they were being used to metabolize and to use as a lipid fuel. So the model was working in vitro, in vivo, and ex vivo. And after having a good model to study the importance and the relevance of the lipids in the brain, we start to analyze what was happening there in our astrocytes. And the first thing that we did was evaluate the oxygen consumption through seahorse. And you can see here in the gray line, these are the regular astrocytes. And the KO ones, they are in blue. And strikingly, we observe how the CPTA KO astrocytes, they have a higher oxygen consumption. They breathe more. And this oxygen consumption was increased basically in the basal state. And that was very, very weird, because we were expecting that if we reduce the fuel in the mitochondria was doing worse. But no, and we did this in all our models, and we got the same result in all of them. So then we wanted to know what was happening in those mitochondria, why they could breathe more. And we know that the mitochondria, they have different conformations to breathe more or less. And this is because the complexes in the electron chain, they are not usually separate as we study in the books. They can be more together, forming supercomplexes. And if they form the supercomplexes, they will be much more efficient. And that we explain that happen in the neurons, that we have the respirosome, we have supercomplexes, and this is very efficient, and it produces low amount of free radicals. In the astrocytes, happens the other way around. We have the complexes less assembled, and we have a lot of free complex one, and this is a little bit less efficient, and it produces much more free radicals that they don't have to be bad. They are just produced. And when we test that as in a technique, is through Western blot, through a native gel, and you can see here how they accumulate the supercomplexes, and the free complex one is almost nothing in the neurons. And if we analyze that in the astrocytes, we have much less supercomplexes and a lot of free complex one. So this was described a few years ago by Irene in our lab. So we wanted to know that this was what was happening in our CPTA KO astrocytes. So we collect the mitochondria from our astrocytes, and we compare the supercomplexes respect the free in the wild types and in the KO. And we quantified that relation, that ratio. And we saw how even if we check the expression of the complex 1 or the complex 3, we have the same effect. We see more supercomplexes in the KO mice respect the wild type. So it was more efficient mitochondria, at least in the supercomplexes. And then we also analyzed the free radicals through two assays, and we saw a reduction of the production of free radicals. So with these results, we saw how these mitochondria that had more oxygen consumption, this was because the supercomplexes were more acomplex than the wild-type mitochondria. So to understand what was happening in the other part of the metabolism, because as I mentioned in the beginning, the glucose in the astrocyte goes to create the pyruvate and then transport to lactate to feed the neurons. But in this case, we have a different mitochondria, so maybe this pyruvate goes inside the mitochondria. And to do that, we label our pyruvic acid, pyruvate, through all the carbons with the carbon 14, and we analyze the CO2 production. And that is the way to see if the pyruvate goes inside the mitochondria to do TCA, a Krebs cycle. And we saw how in the KO mitochondria, we have more TCA, more Krebs cycles, so our pyruvate went into the mitochondria to produce energy, more than in the wild-type case. And also, we analyzed the transformation of pyruvate into lactate. And we saw how this KO mitochondria from these astrocytes, they do less lactate. And to understand which was the key enzyme in this transportation to the mitochondria, we analyzed the PDH-A1, which is a crucial enzyme in this, putting the pyruvate inside the mitochondria. And we saw less phosphorylation in the CPT-A-KO astrocytes, and the phosphorylation implies inactivation. So as we had less phosphorylation, we have this enzyme more active. We analyzed that through the Western, and also we checked the activity of the PDH in vitro, and we had the same result. So we have, in summary, in this astrocytes where we cut the CPTA, and we cannot put the fatty acids inside the mitochondria, we have a remodelation of the complex of the supercomplexes in the mitochondria, and more effective ones. And also we have more glucose transforming into pyruvate and going inside the mitochondria to producing, to giving fuel to the recently assembled chain channel, no? So with these transformations that they were pretty evident and strong, we wonder what was happening with the neurons. Because if we have these astrocytes that they are so different, the neurons, how they are. And we analyze this with two models. One, with the ex vivo system. So in our KO mice, we isolate immunomagnetically, again, with Milten GSAs, the neurons. And also we did a co-culture system. With the ex vivo system, we could evaluate some points, and then to go into the detail, we went to the in vitro system. So what we saw from these neurons isolated from these brains. So here again, we have the wild type in gray, and in orange, the neurons from the KO mice. And we saw how these neurons, they were doing much worse in terms of the oxygen consumption. So they were breathing less. You can see here the regular graphic from the seahorse, and this is the quantification, and it's clear they were doing worse, and they were showing an increase in free radicals. So these mitochondria in these neurons were doing pretty rough, pretty bad. Then we use the co-culture system. So we have in our plate the neurons. In the beginning, just explain the wild type part. So we have wild type neurons, and then we expose these neurons to wild type astrocytes or CPTA-KO astrocytes. And we evaluate what was happening after exposing the neurons to these two types of astrocytes. We analyze the neurons. And here you can see in the first part of the graphs what was happening with the wild-type neurons with the wild types and with the KO astrocytes. And what we saw, it was like the glutathione, so the pentose phosphate product was low in the neurons exposed to this CPTA KO, and we have more ROS production. And the mitochondria was doing a little bit worse because the polarization was lower in these neurons exposed to the CPTA KO astrocytes. You can see the results here, and also these neurons exposed to these KO astrocytes, they were dying a little bit more because of all this increase of free radicals, worse mitochondria, and also the protection against the free radicals was worse. When we analyzed a little bit the function of these neurons, we also saw a decrease in function. So they were dying a little bit and they were doing worse in terms of function. So to understand if we could correct this, or we can like try of see the mechanism behind these problems, we use neurons that they have a tag in the mitochondria. They have a extra enzyme that is catalase, and they have like a extra power against the free radicals. And we did the same assay. We put the mitocatalyse neurons with the KO and with the wild type astrocytes, and we observed what was happening. And what we saw was that the free radical production wasn't increased with the CPTA KO astrocytes. The mitochondria was healthy. There wasn't a reduction of the membrane potential, and we didn't have that increase in the death that we observed with the wild-type neurons. Then we check also the function of these neurons, and we couldn't see any kind of difference. So the neurons that they are extra protected against the free radicals, they were doing much better, and the CPTA KO astrocytes, they didn't do anything wrong for the neurons. So this told us that the mechanism behind the damage in the neurons was mainly because of the free radicals, but it's not excluded. It can be other metabolites. So as we got this, the neurons were doing worse. We wanted to know what was happening in the mice, so we did some behavioral tests, and we analyzed basically memory through two assays. We did a short-term assay, which is the novel object recognition, and then a more working memory assay, that is the Barnes maze. And we saw how with the novel object recognition that you put a new object after training the mice, and the regular mice, they go more to the new object because it's new and they want to see, and they remember the old object. With the wild types that happen, you see here the increase in the discrimination index. That means that the mice goes to the new object because it's new and it's something new that you have to see. And in the case of the KO, this discrimination index goes down. You can see here also the heat map of the say, and here how they go. And in fact, these CPTA KO mice, they have lower memory because they explore less the new object. And then when we test also with the Barnes maze, we saw how these KO mice were doing a little bit worse. They have a lower memory, spatial memory and working memory than the wild type ones. So to sum up, just this is familiar for you. We learn with this work that is already published. You can look for it in the PubMed that when we suppress the CPTA and the fatty acids are not going inside the mitochondria, we have a huge remodeling of the mitochondria, of the electron change. And the supercomplexes, they go all together. They are much more efficient. They produce less free radicals, and that also move and switch the metabolism in these astrocytes because they use more the glucose as a fuel in the mitochondria instead to send the pyruvate to lactate and to the neurons. And what happens then in the whole mice? So these mice, they have a lower cognitive performance. So they are doing much worse in terms of memory. So that is the more important bits of the story. I will be very happy to discuss much more later about it. And just this is, I think, is the most important slide because this is my team. This is my team that they are doing the macrophages story. And this is the team where I do more the regeneration stuff and more the neuro part. So all of us, we are McFadgy, and I would like to thank all the sponsors, especially that there is a network that we are trying to improve the women and minorities in science. So I think they are doing a very nice labor for the science. And finally, thank you for listening me. And I'm very happy to answer all your questions and all your curiosities. Thank you so much.
Thank you. Thank you, beautiful Marina. Beautiful work and beautiful slide. Okay, thank you. So before we go into the Q&A session, let me have an additional introduction. I noticed that our another planet, Professor Pei Shang, have joined our webinar. So let's warmly welcome Pei Shang, and I'll have an introduction for him. Pei Shang, research scientist from the Southern Medical University, China. And he has a very exciting CV and very shining working experience. And Dr. Shang's long-term research interests involve the pathogenesis and potential diagnostic biomarkers and pharmacological management underlying neurological disorders. While in college time, he has been selected as an outstanding undergraduate to visit the University of Manitoba for 2 academic semesters following Dr. Geoff G. Hicks, Director of Regenerative Medicine, to conduct ATAC-seq on a transgenic mouse model of fetal alcohol spectral disorder. And his very shining working experience includes research scientist at the Department of Neurology, Mayo Clinic, USA, and research fellow of Department of Neurology, Mayo Clinic, USA, research fellow of School of Medicine, University of Manitoba, Canada. So welcome, Pei Shang. Okay, after the presentations from our amazing 2 speakers, now let's go into the Q&A session. Okay, welcome all of the professors, scientists, and researchers. Okay, everyone, if you, okay.
Thank you, Dr. Dai, for the introduction. And because I have a meeting around 1 hour later, so I may just put forward some question first. So do you want I put forward questions with each one in separately or just together, which will
It's up to you. So it's up to you.
Okay, I think it's better to discuss separately. So first, because I have done some previous work based on the Alzheimer's disease and alcohol use disorder, so I hope to have some discussion with Professor David. So we have used the ENT1 knockout model, which is adenosine transporter model for the alcohol use disorder. And we have seen that in the ENT1 knockout model, the WAMP1 protein was downregulated in the presynaptic membrane of those mouse in the prefrontal cortex. And also the WAMP1 is a very big compartment for the snare protein, which is important for the synapse activity. So my first question will be very specific. Have you had some work related to the snare protein? Even I know your work mainly based on the tau pathology in the triple knockout mouse, but also I have a question that why you focus more on the phosphorylated tau instead of a better deposition of the AD, because that's kind of like two major hypotheses in AD field. So why you focus more on the tau pathology instead of A beta? And also for my question is that have you looked into some snare protein which is important for the deposition of A beta and tau in the brain of either APP-PSY mouse or the triple genetic editing mouse. And also for another question is it's very exciting to see the DBS applied in this field because DBS is mainly used in Parkinson's disease before, and it's very seldomly applied in the Alzheimer's disease. But my question is there's some difference between Alzheimer's disease and Parkinson's disease. In Parkinson's disease, we know the specific neurocircuits in the human or mouse brain, but in Alzheimer's disease, sorry, in Alzheimer's disease, it's quite complex. We can say the A beta or tau pathology everywhere. So I found you choose the GPI as a kind of region of interest in your study, but I want to know why we can choose other kind of locations prefrontal cortex or hippocampus or other kind of regions. Why you choose a specific GPI in your study, and do you have some recommendations or some comments on this kind of area? So that's my question for Professor David, and I hope to discuss about that.
Okay, well, thank you very much for your question. Nice meeting you today. So several questions. I'll try to answer to all of them. So first of all, well, one very simple answer. You asked me about the snare proteins, which of course are important for the presynaptically for the release correct of the neurotransmitters. No question about that. I didn't really look into that in my models. I was using mainly synaptic markers to see to count synapses, so pre and post synaptically with the colocalization, to see whether there was an improvement in terms of synaptic protein. Also because the proteins that I choose is because they have a local synthesis of synapses. So they have their mRNA there, and they can be synthesized very fast. So for example, in the in vitro model, the stimulation occurs for at least for a couple of hours. So it doesn't last for very, very long time. So I wanted to see whether there was like a kind of a protective response for if the response, the stimulation was protective for synapses by recovering synaptic proteins. So unfortunately, I didn't. It's a very, of course, interesting question what you're asking, but I didn't really look. We didn't really look at the snare complex proteins, which would be important. It would be eventually another step. Then concerning the model that we choose, is because indeed before starting to work on tau pathology, I also work on beta amyloid. And to use the triple transgenic was interesting because we could follow the two pathologies at the same time. So to see the effects of the DBS on A beta and tau. I didn't have the time to show also the data on the A beta, but we published those. It's a paper. I can send it to you without problem later on. But we also saw protective effects against A beta. So we also have reduction of A beta amyloid, especially the beta 42, intraneuronally, but also a reduction of plaques, which was quite interesting, and an improvement into the behavior. So the same mice that we tested for the tau were the same that were also tested for beta amyloidosis and for the behavior. And we could find an improvement in the novel object recognition that Marina mentioned before, and also the Morris-Water maze task. So also beta pathology is of course very important, and I work a lot also on APP homeostasis and A beta 42. There are some publications that I did previously, like in 2009, 10, and 11, on how synaptic stimulation with the same paradigm, using glycine as a co-activator of NMDA receptor, but also in vivo models. I used the TG2576 transgenic mouse models for beta amyloidosis that in one of the slides remained, and I apologize again. But we could see that the protection was the reduction of this A beta 42 with stimulation, recovery again of synaptic proteins, and also an effect on APP homeostasis by basically stimulation induced like a transport of APP more to the cell surface, but also to have more endocytosis at synapses. These are in a 2009 paper that I published on General Science, and I can send it to you. So we have data on beta amyloidose and synaptic activity as well. Now for DBS, concerning DBS, which is of course a very interesting technique that is becoming more and more common for different pathologies. As you rightly point out, we just checked the inner globus pallidus, so the GPI. This is because that was the area that was stimulated in Parkinsonians, because we have the electrodes in the subthalamic nucleus, but that projects to the inner globus pallidus. So that was the area that was expected to be activated, and that is the reason why we only check the pathology in this area. The other areas, of course, they were not stimulated, so we couldn't have expect changes in the tau pathology. We also observe it, for example, in the cortex, but we didn't expect to have changes there depending on the stimulation, because the only stimulated area was this one. Now, in the mouse model, we check in the hippocampus because, of course, we were stimulating in the entorhinal cortex, which via the perforant path goes to the hippocampus, and then all the different areas, so data gyrus until the CA1 and then the fornix. And therefore, in a previous publication, it was shown that when you stimulate the entorhinal cortex, you have, for example, activation of neurons in the CA1 because you see increased, for example, CFOS activity, so immediate early genes, demonstrating like an activation of the neurons in the hippocampus as well. And that is where we also look for the pathology. In the human, as of now, the only samples where we have deep brain stimulation and tau pathology are the Parkinsonians, and the area involved is specifically the inner globus pallidus. Of course, it would be much interesting to also observe, like in the hippocampus or the cortex. Now, of course, I'm in touch with Professor Lozano, so when eventually in time we're going to have access to samples for the subject who got deep brain stimulation in the fornix, then it will be interesting to expand the analysis also in the brain areas involved in Alzheimer's disease. I don't know if I missed any question.
Yeah, that's a good answer for me. Yeah, good to know. Like you have also got the AI data in your previous study, and I'm very interested to look on that. And also, yeah, I know why we have folks like some like GPI data, because mainly the DBS was was performed on the Parkinson patients. Yeah, but now I do know that we have some DBS applied on multiple sclerosis. So in that kind of case, maybe you can get more like interesting brain regions to look at in the human model. But in the mouse model, we can do a lot for that, definitely. Thank you for your response. And also for Dr. Marina, I have very little background for your research. So I will just ask some technology related questions since I have done seahorse in my previous study. So I know it's a very complex and painful process in that of a kind of like a technology. And also, I noticed that you use the primary cultured neurons and astrocytes for your seahorse work. So my question will be very simple. Why don't you just use isolate mitochondria from the mouse brain directly for the like a more mitochondria detection in your work, which is which you can just reflect the uh, aspiration of the mitochondria or the function of mitochondria like directly. Since I noticed you have labeled the pyruvate in your study, so for the isolated mitochondria, maybe we need to add some pyruvate or succinate for the initiation of the oxphos chain. So that would be my question. Why we don't just directly use the isolated mitochondria for that? Yeah, that's my
Yeah.
question. Mhm.
Well, basically, we did primary cells
Mhm.
from astrocytes and neurons, but we also did from the whole brain. We isolated the neurons and the astrocytes immunomagnetically, and then we used those cells from the brain to to see how they breathe. So it was more ex vivo, but it shows very nicely what was happening in the actual brain. It's not exactly the same, but it's better than if we isolated the mitochondria. During the processes, we will lose a little bit of
Mhm.
the reality because, well, it is you have to put the cells into more steps so it will be like equilibrate with the media, and then also you have to add the for the mitochondria isolated, you have to add extra things that if you isolate the whole cell you don't, and it's a little bit more like what is happening in the actual brain. That's why we did some other experiments with the pyruvate and the other things in the primary cultures to check exactly how was working better one complex respect the others, but that was another part of the story. We prefer to isolate the cells and see how they breathe than isolate the mitochondria that the cells will suffer a little bit extra. We
Yeah,
thought that
yeah.
it was better. That is a new protocol that we have used it for the paper, and if you need I can explain you a
Yeah,
little bit
yeah.
how it
That would
works,
be
and it can be done because in our hands works very nicely, and you can see very nice the difference in the different type of cells and see and it's very physiological.
Thank you. And also, I have a very naive question for your work. So you knock out one important gene on from mitochondria, right?
Mhm.
So it should be very lethal for the mice, right? So like the mouse, yeah, I just think about that. So why? Yeah,
Well, the
Still survive?
mitochondria, they, of course,
Yeah,
yeah. No,
still
it's
survive.
we don't do we do a very like acute knockdown as we use the virus. So the mice is already an adult, so we don't have to see how the loss in development of that protein will impact the mice. So that is a bit one way to save these problems with the protein. But although it's a mitochondrial protein and it's very important, it's important for the fatty acid importation into the mitochondria. In general, the mitochondria change and remodelates, but it's still working. So that's why it's not lethal. We didn't leave the animals to live to see how is the span life, but at least the first 9 months, they were more or less okay. They were having problems with the memory, and we also saw some anxiety symptoms, but they were alive and doing more or less okay, and they could eat and they could do the regular things also.
Yeah, that's why I say like in the NRR test and also in the maze test, they just show very bad like NRR scores or just like maze scores. So I just wonder maybe it really like destroys the hippocampus in your mice and causes kind of like a short memory deficiency. Just
Yeah, we didn't test the how was in the specific parts. We just took the whole brain. Well, this is like the first part of a huge story that can go and understand why everything is changing and how everything is so important.
Yeah.
But I thought it was pretty relevant and we got a very exciting results. So we published that and we are continuing working because also it might be related with diseases, how the metabolism, the lipid metabolism impact in the brain can go and can be studied in other diseases, like for example, Alzheimer's or others.
Yeah.
Okay.
Thank you.
Thank Dr. Shang. Okay, how about the questions from our other planets? Professor Wei and our Professor Yan.
Hi. Can I go first?
Yeah.
Can you hear me? Okay. So, very nice to meet you, David and Marina. I have a question for each of you. And first, I mean, both of you give great talks. I enjoy it a lot. So, for my question for David is, I always wonder about the specificity of DBS treatment because it's already used in the clinic, and you have to implant it in a certain brain region. I wonder, like in mouse models, what is like the brain region specificity of the DBS effect, especially on the effect of DBS that you observe on the synapse activity. So brain region specificity is my first question. And then how about cell type specificity? Because you're looking at synapse function. So, there are excitatory neurons, inhibitory neurons, or, maybe we can be more specific, like do you see any difference in the effect of DBS on like excitatory synapses versus inhibitory synapses? So that's my question for David.
Yes, hi. Nice meeting you, and no, thank you for your questions, which are of course very, very important. So, in mice, in fact, we know the idea was to activate the hippocampus or like this circuit. So now there were experiments that were done in rats, and in rats, I mean, the brain is a little bit bigger, so it was possible to pinpoint more the fornix in a more specific way. The mice, the fornix is very, very small, so that's why we went for the entorhinal cortex, which will activate all the hippocampal circuit as well. Now, concerning the specificity of the neurons, the subpopulation of neurons, that is, of course, a very important point. So we know that, at least for the hippocampus, typically, if you stimulate neurons in the entorhinal cortex, you via the perforan path, you activate mainly the glutamatergic neurons, excitatory neurons, or the glutamatergic system that will then activate the hippocampal circuits via the mossy fiber and the shaft collateral. Now, the idea that is going to be mainly the excitatory neurons, glutamatergic, is also given by our in vitro data, where we have consistent results basically having like hippocampal neurons and induce a stimulation activation of NMDA receptors. So that would be given by excitatory glutamatergic input that we have. That is true for the in vivo and eventually for the mouse model. For the human, of course, the situation is much more complex, especially when we go, for example, to the Parkinson's disease, because it is true that, I mean, discussing with Professor Lozano, but with others, is not still 100% clear of what exactly happens in the brain, meaning that sometimes you can also just reduce activity of inhibitory neurons, for example. I mean, I was looking into that, and sometimes you can also have sorry, I'm trying to find, because I put into the discussion of my last paper this interpretation. I was trying to see exactly what was because, as you say, I mean, the deep brain stimulation is pretty complex. So even the surgery themselves don't really know specifically what are the population of neurons. I mean, they know that there's a nucleus that is activated or inhibited, but what is the activity that then occurs? It's a little bit tricky. There is one idea is that either can stimulate the neurons that are postsynaptically, or it can also reduce the firing patterns that is altered usually in neurodegenerative diseases. For example, in Parkinson, is also known that you can have, by a compensatory mechanism, you can have a higher firing rate that needs to be re-equilibrated. It can also give origin to other kind of, besides the tremor, there is also dyskinesia involved. So it's of course a very good point. As far as we know, in our model, and also for what happens in the brain of the patients now treated for the brain stimulation of the fornix, seems to be mainly like the excitatory neurons that are the one involved. But of course, we cannot exclude also what happens to the inhibitor. That would be very interesting to investigate, of course.
Thank you. Thank you very much. And my question for Marina is, so your research is very interesting because that's also related to my own work. You know, I also work on astrocyte and lipid metabolism, but in the context of epilepsy patients and mouse models. So my question is, so in your work, you saw that if you knock out CPT1A in a mitochondria of astrocyte, under physiological conditions, you can inhibit the fatty acid oxidation, you reprogram the metabolism, and you can affect the pyruvate lactate shuffle to neurons. I mean, which is like very solid observation, but I'm like more interested in what is the potential implication of this physiological process to like neurological diseases. You know, to your knowledge, are there any neurological disease conditions on which is associated with like defects in the function of this gene, or you know, the related mitochondrial fatty acid metabolism pathways that may be implicated in a certain disease, or maybe it can imply that we can manipulate the function of this gene or this pathway to help treat any disease or anything like that. That's my question.
Okay, exactly. Yeah, I have my mic on. So exactly, I don't know if there is a mutation in CPTA that will show neurological problems. I don't know. I'm not aware. Well, we were basically centered in the mechanism and very basic stuff, but what we saw with our results that might be very interesting is that in the we did the metabolomics of the whole brain, and we saw like I think it was an increase of GABA And well, I remember explaining my story, and one college came and he told me, why you didn't test this for Alzheimer's? Because we got that paper that explained that GABA is very important in Alzheimer's. So that put us a little bit in the clue of maybe there are other diseases that they are already established that they have problems in the lipid metabolism, but we didn't have the chance to go further and to examine all the metabolites that we saw problems and try to mix that with our research. But we are very open to study this kind of problems in metabolism with the disease models, because I'm sure there will be very strong connections there.
Thank you.
Thank you so much.
Okay, questions from our Professor Wei. Have any questions about the two presentations?
Thank you, Professor Dai. This is Dr. Wei from Beijing, and I have a question for Professor Templiner. Thank you for your interesting presentation. So you introduced DBS as a option to modulate tauopathy, and you introduce much content about AD, Alzheimer's disease. But clinically, DBS is hardly a choice for AD patients. This is not like Parkinson's disease. So why you choose DBS as a treatment methods? In clinical practice, we can use TMS, TDCS, TACS, such as this non-invasive methods. Do you think these non-invasive methods can show similar effects on tauopathy? Thank you.
Yes. So hello, well, nice meeting you, and thank you for these very interesting questions. Absolutely. So why I use DBS? Well, because actually it was a collaboration that started a few years ago with Professor Lozano. He's a main expert in deep brain stimulation in humans to treat different pathologies, and it was like a kind of serendipity way by which he decided to use DBS for Alzheimer's disease. So already, I mean, in Alzheimer's disease, there was another kind of DBS approach that was used for the nucleus basalis of minor that was not really giving positive results. So this one was because Professor Lozano told me, actually even put into a review, that he was making a surgery on a patient who had for morbid obesity, and you know that the patients are awakened, and at a certain point he started to describe memories that were becoming more and more strong, memories that he thought he lost of many years before. And so when they went to check what was the area that was activated, mainly, they found that during this procedure, they found that the fornix was really firing. So that was the link with the memory boosting and eventually therapeutic for Alzheimer's disease. Now, I'm not a surgeon, a neurosurgeon, but I'm a cell biologist, and my point is really to try to understand in details, as I'm trying to do, what are the molecular mechanisms that are triggered by deep brain stimulation or by synaptic stimulation. So the goal for future therapeutics would be indeed to find either like a molecule, a method that can trigger the same pathways that are driven by the electrode and the deep brain stimulation, that will help the neurons to regenerate or become healthy again. Eventually to boost also neurogenesis, because that's also what they found with DBS in the hippocampus of mice. That there was like an increased neurogenesis. Yeah, so DBS is one approach that I found interesting, and there was this very strong collaboration. But of course, I believe that there are other methods that can bring to the same results or induce the same positive outcomes that we don't know yet. I mean, of course, there was also this method of stimulation via kind of ultrasound of like external devices without having an implant, of course. The problem that was that the patient was supposed to go, since they are very big devices, they cannot be at home. So the person is supposed to go to the hospital pretty regularly to do this, which is, of course, like a downside, rather than to have an implant that you can regulate. And now, you know, that there are software that can regulate like the frequency and the stimuli that you have that are like custom made for each patient, especially for Parkinson's disease, that you can do even almost at home. So that is, let's say, more convenience for the quality of life. But I mean, I think these are important progresses, and especially it's important to, from my point of view, to understand what happens really to the metabolism, not only of neurons, but eventually also to glial cells, which are equally important, and figure out new targets or now new therapeutic avenues than just, of course, like a surgery that is a very invasive method. So I don't know if I answered your question, but it's, of course, an important topic.
Thank you very much. Thank you very much. So I have another question for Dr. Gracia Moshier. That's the right pronunciation. So you introduced fatty acid oxidation. That's a very important mechanism. So my question is very similar to Professor Yan. So just ask you. So in the view of a clinical practice, so how do you think yield finding can be used, for example, to predict the risk of Alzheimer's disease, or yield finding, whether yield finding can be used to develop a method to treat Alzheimer's disease. So you know a biomarker called GFAP. So abnormal GFAP level is this is related to exercise. So and abnormal GFAP level can predict the risk of Alzheimer's disease many years ago. So what do you think the potential for prediction or treatment for Alzheimer's disease of your finding? Thank you.
Okay, thank you so much for the question. It's a very tough one. Yeah, it's difficult. Well, I read also the news about the GFAP as a predictor of Alzheimer's, and I found it very interesting. That underlines that the glia in the diseases are much more important than we thought in the beginning. But as a result of our research, I don't know if I can predict that some of our markers or we can find a marker to see Alzheimer's yet. We might need to, well, I would suggest to do like genetic screenings to see if there is a mutation of CPTA and see if that can be a predictor. I don't know yet if we can get something else from what we have until the moment, because I guess we should study how is the metabolism with Alzheimer's models before to reach or jump to a conclusion. And as a therapy, I know that ketogenic diets are used for some of the some neurological disorder, but I don't know I don't think that it will work for Alzheimer's. I don't have enough information. So I guess we should study more and we should test our results in disease models and see how they are. I think that is what we got is very interesting, and we need to jump to the diseases and see what is happening there in the pathological. So we might establish a collaboration with some of you to test in your disease models and see what we can find. And maybe in few years we can get a predictor for the disease. So thank you
Thank
so
you very
much.
much. So we may discuss later. Thank you.
Okay. Thank our two amazing speakers for giving us such perfect presentations. Thank our very shining planets, such as Professor Yan, Director Wei, and Dr. Shang, provide us such a heated discussion. And last but not least, we should thank the Journal of NRR provide us such a beautiful morning, afternoon, or evening. So we hope we can continue our communication deeply in the future, even face to face. Okay, all the best. Thank you, everyone. Okay, bye-bye.